In eukaryotes, the primary RNA transcript of a gene needs further processing before it can be translated. So in eukaryotes, that messenger RNA that is produced cannot start translation immediately. It has to be processed. And that term process means several things must happen to that messenger RNA before it becomes a mature messenger RNA and start the translation process. All right, we'll get into the details of what RNA processing involves. So this step is called RNA processing or transcriptional modifications. Also, it needs to be transported out of the nucleus into the cytoplasm. So take note, in the case of prokaryotes, there is no nucleus. So that genetic material is found floating in the cytoplasm along with other organelles such as the ribosome. So when that messenger RNA is formed in prokaryotes, it can easily find its way over to a ribosome and start translation, even when transcription hasn't finished. In eukaryotes, it's a different scenario. Has to wait until that messenger RNA is finished. Thereafter, that messenger RNA must go through what is called RNA processing or post-transcriptional modifications before translation can start, okay? Ribosomal RNA links amino acids together to form proteins. This occurs in the ribosome, right? The cap at 7-methylguanosine, which helps to protect from attack by ribonucleases. This what we're explaining here are the process, but this will be explained even better with my diagram on the next slide. So one end of that messenger RNA is capped with a methylguanosine, and it helps to protect from attack from ribonucleases. Splicing. When you hear this term splicing, what do you think this means? What do you think splicing means? It's not slicing, all right? It's splicing. What does that mean? Splicing actually means to join. All right, so to join. So you're gonna take one piece, one end of this molecule and this other end from this molecule, and you join it. That is splicing. So splicing is catalyzed by large protein complexes called spliceosome, assembled from proteins and small nuclear RNA that recognize the splice sites. So remember we said that small nuclear RNA is only found in eukaryotes, not in prokaryotes. And so one of the roles of small nuclear RNA is to recognize these splice sites and know exactly where these molecules should join. Poly-A tail protects the 3' end from ribonuclease digestion. So these are the steps that are involved in RNA processing, and you'll better understand it from this diagram, okay? So here we have at the top a gene. All right, so this is a gene. And you take note that the gene is comprised of exons and introns. All right, remember this is in eukaryotes. So eukaryotes contain introns and exons. Okay, so that's a gene. Transcription takes place. And after transcription, we end up with what is called a pre-messenger RNA. Now this process has been completed. All right, so transcription is over, and we end up with what is called a pre-messenger RNA. All right. Now there are several things that must happen to that pre-messenger RNA before translation can start? The first thing is capping. All right, so capping of this 5' N, and it's capping with what? It's right there in the text. All right, capping with 7-methylguanosine, and the purpose of that is to help protect that 5' N from digestion or from ribonucleases. Those are enzymes that can actually degrade that N. So that is the other step, capping of this 5' N with 7-methylguanosine, okay? The next step in RNA processing is excision of introns. Excision means cut, right? So you actually cut these introns right here, 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 and here. So these portions are excised from these introns. And splicing of exons. Remember what splicing means? To join. To join. So take note of what happened there. The introns are actually cut out, because remember we said that introns play no role in terms of gene expression. So introns play no role. So they have to be cut out. So the introns are excised and the exons are spliced. And that is what we have here at this level. All right. So if we notice, we have our capped 5' N and we have only exons. The introns have gone. Those have been excised. The last step in RNA processing is polyadenylation. So it's a capping of the 3' N with this poly A tail. And that helps to protect this N from digestion from ribonucleases as well. And so after we have our exons only, our 5' N that has been capped, our 3' N also has been capped, we end up with what is called a mature messenger RNA, which is then ready to go to the cytosol for translation by ribosomes. All right? So folks, this, what I'm explaining, takes place only in eukaryotes. Remember we said that for splicing of exons to occur, the small nuclear RNA plays a key role in recognizing splice sites. And that is the reason why small nuclear RNA are only found in eukaryotes. I also said a while ago, prokaryotes do not have introns. And hence the reason why they don't have to go through this processing. And that is the reason why translation can start even before transcription is even over. Is that understood?
So you could repeat the last piece again where you said from transcription, it begin before transcription, sir?
Okay. This what I just explained is called RNA processing, and this takes place only in eukaryotes, simply because eukaryotes have introns that must be cut out. All right? So after this process of excising introns, then the exons must be spliced, must be joined together. And small nuclear RNA plays a key role in recognizing the splice sites. Okay? So I mentioned that RNA processing is required in eukaryotes because eukaryotes have introns. In the case of prokaryotes, bacteria do not have introns. So their messenger RNA don't need to go through this process. And for that reason, because they don't have introns and don't have to go through RNA processing, for that reason, translation can start even before transcription is over. Is that understood?
Okay, sir, thanks.
All right, you're welcome. Any other questions? Wilbur?
Sir, so the result for prokaryotes would be a polypeptide, and then in eukaryotes, the mature mRNA molecule, right?
Can you talk a little bit louder or go closer to your mic, Wilbert? I'm hearing you but very low.
Sir, I was saying if there is the end product for prokaryotes would be a polypeptide, whereas in eukaryotes the end product would result in a mRNA molecule.
I think what Wilbert is saying is that after transcription in prokaryotes, you end up with a messenger RNA that goes to the ribosome. In the case of prokaryotes, translation starts even before transcription is over. So before transcription is over, it is already forming that polypeptide. In the case of eukaryotes, what we see here, after transcription, immediately after transcription, translation cannot start yet because that pre-messenger RNA has to go through processing. So it has to wait until you obtain a mature messenger RNA whereby the ends have been capped and the introns have been excised. All right. So in eukaryotes, once you end up with this mature messenger RNA, what we see here at the bottom, then this will find its way to the ribosome where it will start building that polypeptide. Okay. I'm not sure if that answered your question, Wilbert.
Sir, yes, sir.
All right. Are there any other questions with respect to RNA processing? And so please understand why RNA processing occurs in eukaryotes and not prokaryotes. All right. So prokaryotes do not have introns and hence do not need to go through all this processing. While eukaryotes, because they contain introns, they have to go through this processing to cut out those introns and produce that mature messenger RNA. And only after that is produced, then translation can start. Okay? Any other questions? All right, I will show you guys a video, and the video is on transcription. It will explain all of this transcription in prokaryotes as well as transcription in eukaryotes. All right, why is it necessary that RNA processing takes place in eukaryotes? All right, and then after the video, we'll continue and move on to translation.
To do in this video is a little bit of a deep dive on transcription. And just as a bit of a review, we touch on it on the video on replication, transcription, and translation. Transcription in everyday language just means to rewrite something or to rewrite some information in another form. And that's essentially what's happening. Transcription is when we take the information encoded in the gene in DNA and encode essentially that same information in mRNA. For transcription, we are going from DNA. We are going from DNA to messenger RNA. And we're going to, in this video, focus on genes that code for proteins. So this first step is the transcription, DNA to messenger RNA. And then in the future video, we'll dig a little bit deeper into translation. We will translate that information into an actual protein. But these diagrams give a little bit of an overview of it. It's a little bit simpler in bacteria. You have the DNA just floating around in the cytosol. And so the transcription takes place. You start with that DNA, that protein coding gene that is the DNA. And then from that, you code the messenger RNA. You see that in that purple color right over here. And then that messenger RNA can be involved with the ribosome. And that's the translation process to actually produce the polypeptide, to produce the protein. in eukaryotic cells, and we're going to get into a little bit more depth in this video, the transcription, the DNA to mRNA, that happens inside of the nucleus. And there's essentially two steps here. You go from DNA to what we would call pre-mRNA. Let me write that down. Pre-mRNA, which would be depicted right over there. And then it needs to be processed to turn into what we would call mRNA, which then can leave the nucleus to be translated into a protein. So now that we have that overview, let's dig a little bit deeper into this and understand the different actors and understand, if we're talking about a eukaryotic cell, what type of processing might actually go on. So right over here, we are going to start with the protein coding gene inside of the DNA, right over here. And we are going to, the primary actor that's not the DNA or the mRNA here is going to be RNA polymerase. It's used to create a sequence that will become a nucleotide sequence that will become the messenger RNA. So this RNA polymerase, it needs to know where to start. And the way it knows where to start is it attaches to a sequence of the DNA known as a promoter. And every gene is going to have a promoter associated with it, especially if we're talking about eukaryotic cells. Sometimes you might have a promoter associated with a collection of genes as well. But in general, if you've got a gene, you're going to have a promoter. And so that's how the RNA polymerase knows to attach right over there. And so once it attaches, well, then it is able to separate the strands. It separates the strands. And it's pretty interesting because when we went deep into replication, you saw all of these actors, the helicase and whatever else, but this RNA polymerase complex is actually quite capable. Not only it separates the strand, and then it's actually able to code for the RNA. And it does that the same way that when we study DNA polymerase, it does it in only one direction. It can only add more nucleotides on the 3' end. So it encodes from the 5' to the 3' direction. Notice this arrow here, we're extending it on the 3' end of the RNA. And so as you can see here, when it does this, it's only encoding one side of, or it's only interacting, I guess you could say, or coding complementary information to one side. Let's think about this a little bit. You can call the side that it is forming, that is interacting with, you call that the template strand, because that side of the DNA is acting as the template for forming that RNA. But if you think about the information that RNA is actually going to encode, well, it's going to contain the same information as the coding strand of DNA, the other strand of DNA, because these nucleotides right over here, this nucleotide is going to be complementary to this one over here, just as this nucleotide was complementary to that one over there. And you can see it in a little bit more depth if we actually were to add the nucleotides. So this is the template strand. If you have thymine, well, on the RNA, you would have the adenine. And look, on the coding strand of DNA, the one up here, you would also have an adenine. And they are essentially, the coding strand and the RNA essentially end up being the same sequence, but the one difference is that you won't find the thymine in the RNA. Instead, you'll find a similar nitrogenous base, and that is uracil. But uracil plays the role of thymine, so you're essentially coding the same information. So once again, this bottom strand is acting as a template, but it's going to be the resulting RNA that gets coded is essentially going to have the same information that we had in the coding strand. And just to get an appreciation for what this looks like, and I would even write, I'd put looks in quotations, I even did little things with my fingers when I said that, is that, it's hard to really visualize what these things look like, but you can see here that the RNA polymerase complex, and this is for a specific organism, can be very, very complex and involved, and it's fascinating how these things interact. And every time you're studying biology, and someone like me is going to give you these nice, clean narratives of how these enzymes interact with the different macromolecules like the DNA or the RNA, you should always remember, this is amazing. These are these molecules interacting with each other, bouncing into each other. It's happening incredibly fast inside of the cell. You should be in awe of this. It's happening in all of your cells, or as we speak. unique. So this is pretty incredible stuff. So the next thing you have to think about, this right over here, we are extending the RNA. Well, when does this thing actually stop? And it stops once so this RNA polymerase is going to keep going on, and then it's blue where we've labeled this a terminator. Right? This area is a terminator, and there's multiple ways that signals to the RNA polymerase that, hey, it's time to stop, or more particularly that it somehow creates something structurally that the polymerase just lets go. One mechanism that's depicted right over here is that the mRNA that's coded, and this is typical, or this can happen in bacteria, is that the mRNA that's coded forms a hairpin. So it has to have the right complementary base pairs right over here to form this hairpin. But this hairpin, along with the things around the hairpin, essentially make it impair the polymerase to keep on going. And so the complex kind of changes a little bit, and so it lets go, or at least that's how people believe it. There's other forms of how the terminator can act. It might be sequences that parts of the polymerase complex recognize, and it makes a conformation change so that the RNA polymerase lets go. Now, if we're talking about a prokaryote, we're done. We would have formed, this will be our messenger RNA, which then can go to a ribosome and then be translated into a protein. But if we're talking about a eukaryote, well, then we have to do a little bit of processing. If we're talking about a eukaryote, if this is a prokaryote right over here, this would be our mRNA. If this is a eukaryote, then this is our pre-mRNA, which now has to be processed. And you might say, well, how is that going to be processed? Well, there's a couple of things that are going to be done. Some things are going to be added at the beginning and the end of the mRNA. The 5' cap, this is a modified guanine. Modified guanine right over here, which is going to help in the translation process as the ribosomes attach onto it. And then you have this poly A tail, and it's called a poly A tail because it has a bunch of adenines at the end right over here. And these not only help in the translation process, it helps make sure that the information is more robust, that the ends of the mRNA don't in some way become, or it makes it less likely that they're going to become damaged. Now, the other thing that needs to be processed, and this is one of those fascinating things in evolutionary biology, is that we will have in this mRNA sequence, you're going to have parts of the sequence which we currently consider to be nonsense sequences, and we call them introns. And I'm going to put it in quotes because in general in evolution, it's seldom that things have absolutely no purpose, but these are not coding for the protein that is going to be coded by our initial gene. And so these are actually processed out. They are spliced out. And I'm not going to go into all the details of the actors that cause the splicing, but as part of this eukaryotic processing, you add the cap, you add the tail, and then you splice out the introns. And then once you spliced out the introns, all you have left are the exons. So you have that, it's going to be connected to that, connected to that, and so this is what you have resulted. This is in a eukaryote. You will have this mature mRNA, and that's what we saw. That's what we saw right over here. That can then, let me underline that right over here, which then migrates out of the nucleus to a ribosome where it can be translated.
All right, any questions on transcription? All right, it's all good, right? Now let's move on to translation. All right, so translation occurs where? In which organelle? Translation occurs in which organelle, guys? Ribosome. Okay? Ribosome. So the genetic code, the conversion in the cell of the messenger RNA-based sequence information into an amino acid sequence of a polypeptide is called translation. All right? So remember, the RNA message is taking a code from the DNA molecule, all right? And then it will take it to the ribosome, whereby it will translate that coded message into a sequence of amino acid, all right? The DNA base pair information that specifies the amino acid sequence of a polypeptide is called the genetic code. Let's take a look at the nature of the genetic code. Messenger RNA molecules are read in groups of 3 called codons to specify the amino acid sequence of a protein. So, if we have an RNA molecule here, all right, so A, G, C, U, G, A, C, G, C, A, G, A, U. Remember no T in this, right? Because it's an RNA molecule. C, U, A. What it says is that these will be read in groups of trees called codon. So this here will be one codon. And these codons code for specific amino acids. We'll come to that in just a few. So this here is another codon here, another codon, and so on. All right? So it would read the message in groups of trees, and these are called codons. And each codon code for a specific amino acid. So you'd have an amino acid here, an amino acid here, one here, one here, one here. And this is how this polypeptide is being built, right? These are joined together. Okay? So it is messenger RNA question. Somebody had a question? All right. So it says messenger RNA molecules are read in groups of 3 called codons to specify the amino acid sequence in protein. 4 different nucleotides, A, C, G, and U, occur in the message, and 20 different amino acids occur in proteins. If it were a 1-letter code, then only 4 amino acids could be coded. All right, so if it were a 1-letter code, we only have 4 letters, then that would only code for 4 amino acids. If it were a 2-letter code, then it would be 16 amino acids. A 3-letter code, like what it is, a 3-letter code, would generate 64 possible codes. And we will take a look at these codes in just a while. All right, so since we're talking about groups of trees, all right, 3-letter code, it will generate 64 possible codes. Some amino acids may be specified by more than 1 codon. So you have more than 1 codon coding for the same amino acid. All right. I'll show you a table with the different codes, genetic codes, and the amino acid that they code for, and you'll understand what that is saying. The characteristics of the genetic code are as follows. The code is a triplet code. meaning that it comprises of 3 bases at a time. So every 3 base, all right? That is 1 codon. The code is comma-free. So the messenger RNA is read continuously. 3 nucleotides, which is equivalent to 1 codon at a time, without skipping any nucleotide of the message. So it is continuous. It will not omit any of these bases. So it will run continuously. As soon as it reads the first 3, then it reads the next 3, then the next 3, and so on. It will not miss any of the bases. The code is non-overlapping, so it will not overlap. So here we have the first codon, and here we have the second codon. It will not overlap and say, all right, it will read this one now. No, it will not overlap, all right? The code is almost universal, so all organisms share the same genetic language. The code is degenerate. What does that mean? The code is degenerate, with 2 exceptions, which would be AUG. All right, this codon, which codes for this amino acid, methionine, and this code, UGG, which codes for this amino acid, tryptophan. So with the exception of these 2, more than 1 codon occurs for each amino acid. This multiple coding is called the degeneracy of the code. All right, so the degeneracy of the code has to do with multiple codes code for the same amino acid. Okay. The code has start and stop signals. Methionine, all right, which is AUG, is most commonly used as the start, as a start codon for protein synthesis. So all the time you will have that particular codon being the first one. That is the start codon in protein synthesis. 61 of the 64 codons specify amino acids. So 61 of the 64 codons specify amino acids. These codons are called the sense codons. The other 3 codons, which are UAG, which is for amber, UAA, ochre, and UGA, which is opal, these are called the stop codons or non-sense codons or chain terminating codons. All right? So remember that you have one that is usually the start codon, that is methionine, AUG. All right? So that is included in the 64 that codes for amino acids. And then you have these 3 codons, which are the stop codons, which are usually at the end of the translation process. Okay? Chain terminating codons. Here we have a list of all 20 amino acids. They're in alphabetical order. And it shows the abbreviations and so on. I'm not expecting you guys to memorize and know all of these. All right? This you'll have at your disposal anyway when doing an evaluation. So if I ask you to build, I give you a DNA molecule and I tell you to build a protein from there, then you'll have to first build your messenger RNA, and from your messenger RNA, find which code corresponds to these amino acid, and you build your protein. All right? Of course, it has to be in the particular sequence that these codons appear on. All right, so you have, I'm sure you've heard of most of these amino acids before. All right, so these are all 20 amino acids. This table here shows the genetic code. All right. Remember, we said that it's a triplet code. So if we look at this code right here, UUU, UUC, UUA, and UUG, all of those codes code for the same these amino acids. For example, the first two would code for this amino acid right here, and then the bottom two would code for this amino acid at the bottom. Okay, we mentioned the start codon, which is AUG. This codes for this amino acid, methionine, that is usually used as the start codon. And then we also mentioned the three stop codons that we see up here. So UAA, UAG, and UGA. These are usually the stop codons. So that would be at the end of the translation process. So when we refer to as the degeneracy of the code, all right, you take, for example, here, at the bottom here, these four codes code for the same amino acid, valine, all right? That is what the degeneracy of the code means. So if on that RNA molecule, all right, this is the RNA molecule, and you see it says G, U, U, all right, and then G, U, A, and for example, U, U, U. All right, we're talking about three different codons here. Of course, this is this amino acid. This is another valine. And then this one would be this other amino acid. And so you can build your polypeptide. This will be your polypeptide being built based on the information from your messenger RNA. Because the messenger RNA is bringing that coded message. All right, and the message is read in groups of 3 nucleotides, which are referred to a codon. So there we have 3 codons, which would represent 3 amino acids. All right, is that clear? So I can give you a DNA molecule. All right, I can give you a DNA molecule. Of course, it would have the base sequence. And I can ask you, well, it will be a particular gene. And I can ask you which protein will be built, or mention the sequence of amino acids that corresponds to that particular gene. Is that understood? Of course, you will have this table at your disposal, this genetic code table, whereby you can see the different codons and see which amino acid it corresponds to. Pretty simple and straightforward, right? Questions?
Sir,
Go ahead, Wilbur.
sir, so each code is written as it appears in the mRNA and should read in the 5' 3' direction, right, sir?
Yes, that's correct. So each one of these codes here are the codes that will be read from the messenger RNA, reading from the 5' to the 3' direction. Any other questions? Of course, you'll have this table at your disposal as well, whereby you see the different amino acids. So you'll have this. So you'll know the different amino acids. You'll also have the genetic code table that indicates these codon and which amino acid they code for. Questions? You can ask the question now before I ask the question next week Thursday, because next week Thursday you'll have test number 2, I think. And it will cover materials from that was evaluated on the last quiz, as well as what we discussed here in terms of the central dogma of molecular biology. So it will cover replication, transcription, and translation. You can ask the questions now before I ask the question next week Thursday. All right, so it's pretty straightforward, right? So I can give you a DNA molecule. And I can ask you what polypeptide will be built from that molecule. Clear? All clear?
Yes, sir.
Okay, it sounds like people are scared already. People don't even want to talk. All right, so let's try and get an understanding of exactly what takes place during translation, right? In the ribosome. So here at the top, here we have the ribosome. This is the organelle floating in the cytoplasm. So all of this here is a ribosome, okay, that you see there in blue. And you see the messenger RNA coming in, actually coming in. And it comes with a coded message with these letters A, G, C, and U in different order, different sequence. Okay? So in the ribosome, there are 3 sites where these transfer RNA, all right? So if you notice these transfer RNA that we see on the right, that has the shape of a cross, these will be bringing in the corresponding amino acids. All right, so here, the first area where these transfer RNA will enter the ribosome is called A site, all right, or the acceptance site, because there is where it will accept the ribosome, the amino acid, sorry. Then the second site is the peptidyl site, where it will be adding these amino acids. You see what is happening here? This is a growing polypeptide chain. All right. And after it releases the amino acid to the growing polypeptide chain, then this transfer RNA will exit the ribosome from the exit site. And so these, what we have here on the left, would be empty transfer RNA. It means that they have done their job in terms of bringing the corresponding amino acid to the ribosome to build up polypeptide, and they exit without the amino acid. So they're released from the amino acid, and now they can go and pick up other amino acids. So here we have, from the right, these transfer RNA. They're coming to the ribosome, bringing with them these amino acids. And take note, the amino acid corresponds to the codons on the messenger RNA. All right? And so what these transfer RNA have is anticodons that corresponds to the codon on the RNA molecule. So let's take a look at this one, this codon, for example. This one right here. The actual codon is GUG. That's the codon on the messenger RNA. And it codes for which amino acid? Which amino acid does GUG codes for? It's right there in the diagram.
Valine.
Valine. See it here at the bottom. So GUG codes for valine. All right? But this transfer RNA has that anticodon that had to match that codon, which is CAC. But the codon that is on the messenger RNA, that is the one that will indicate which amino acid is it. So remember this, GUG, all right? So GUG. And we said that that code for valine. Let's go to the genetic code table. GUG, remember that. Let's find GUG. It's right here at the bottom. GUG. All right, GUG, and it codes for which amino acid? Valine. Okay, so the coded message that will correspond to the amino acid is the one that is on the messenger RNA, not the anticodon that is on the transfer RNA. Is that clear? So that is what you need to focus on, the coded message that is on the messenger RNA. And from there, then you can go to your genetic code table to find out which amino acid corresponds to these particular codons. All right? So this right here is the polypeptide chain that is being built when these transfer RNA are bringing the corresponding amino acid. And here you have this growing polypeptide chain. All right? And that is continuous until all the coded message on the messenger RNA are read and translated. All right? Questions? I have a couple of videos that I will show you that hopefully you'll better understand translation and what this diagram is depicting, all right, of these transfer RNA bringing the amino acid, all right, and building that polypeptide chain. All right, so I'll show you guys two videos.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, so there you have it. That's the information that will be evaluated for test number 2, which will be next week, Thursday. I think I will open it from, was it from 4 o'clock that I used to open it? 4 o'clock in the afternoon and close at 11?
Yes, sir.
Okay, so that's the case for test number 2. Remember, it will be, the content to be evaluated will be what was covered on quiz, the last quiz you did, including this portion which has to do with the central dogma of molecular biology, replication, transcription, and translation. All right? Questions? None? All right, folks, Lourdes, go ahead.
The last thing, sir, would we be taking our quiz before the test?
The quiz that you missed?
Yes, sir.
I'll have to try and check my schedule and see if I can get that in to you guys probably, I don't know, tomorrow or before the test. I'll see.
All right, thank you, sir.
Okay. All right, folks, if there are no other questions, then take care, enjoy the rest of the week, have a great weekend, and see you back here next week, Tuesday. Take care.